Hello friends, Ace Botany next episode. In this episode, we'll be discussing principles of inheritance and variation, sixth one. So for convenience, I will share the screen. I think screen came. So shall we proceed? So what all topics we'll be discussing in the sixth part of principles of inheritance and variation? 5 main topics with the sixth questions, theory and runs with the key. So first topic is sex determination general. Second is chromosomal or genetic basis of determination. Third is heterogamity, male, female. Fourth is honeybee, special sexual determination method. Fifth is sex-linked inheritance. And sixth, as usual, question. So these are the topics we'll be discussing. So we can proceed. See, when we discuss principles of inheritance and variation, sixth one, it deals with the sex determination. So what is or when the sexual reproduction first appeared or came? Scientists believe that sexual reproduction first engaged by some primitive protistants. Single cell protistan means single cell, almost around 2 billion years ago. Some suggestions are there, some primitive algae and bacteria sexuality came early. That is regarding the origin of sexuality. Now, why sexuality developed? Sexuality developed, it is an adaptation for genetic recombination, leads to adaptation to meet competition, to produce variation among offsprings for survival. So, when it came, why it came? Now comes, what is sex determination? See, it is the event that determines an individual will develop into male or female. In this world, different species have different methods for sex determination. The intrinsic and extrinsic factors also have a role in it. Thus, we can find different criteria available for sex determination. Most acceptable mechanism is chromosomal or genetic basis, this one, of sex determination. The basis of this type of study was given by famous scientist Hermann Henking. This is the person. Lifespan is 1858 to 1942. And his discovery done in 1891. It is a usual question for theory and runs. He was able to notice a specific nuclear structure in spermatogenesis of certain insect. Spermatogenesis. He was able to identify only 50% of the gamete or sperm received that particular nuclear structure. So he given a name as X body. But what is the need? What is the significance that he was not able to suggest? Later, scientists identified this X body given by Hermann Henking is nothing but present X chromosome. I think that idea is clear. Now, Y chromosome discovered by Stevens. Similarly, X and Y chromosomes are sex chromosome by Wilson and Stevens. Now we have to proceed to heterogamity. First identify two types, then we will be explaining what is heterogamity, etc. Two types of heterogamity we have to proceed. One is male heterogamity, other is female heterogamity. Now what is meant by the term heterogamity? See, it is the process of producing two different types of gametes. one will produce male offspring, other will produce female offspring. So two types of gamete. One type of gamete, male offspring will be produced, other type will be producing female offspring. So first we have to start with the male heterogamety. What is the meaning? Meaning is males are able to produce two types of gametes. That's the meaning, simple meaning. Better identify what is the meaning of it. Then if we proceed, it will be easy. I think first page is clear to all of you. We can proceed to second page. So when we come to male heterogamety, first is here we can find X0 male, XX female. That means parents are AA plus X0 diploid. Here AA plus X0. So female is homogametic, male is heterogametic. And how many types of gamete from male? From male, gametes will be A plus X, A plus 0. So two types. Females are only one type, A plus X. Now these two will be coupled, these two will be coupled. So A plus XX, that is female, A plus X0, that is male. That is how the mechanism. So this is a typical example for X0 male, XX female. Sex determination example is grasshopper. I think first idea is clear. Now male heterogamety is second type. XY male, XX female. As usual, parents will be AA plus XY, AA plus XX. Gametes will be AX or A plus X, A plus Y. Here only one type of gamete, A plus X. So these two will be coupled, these two will be coupled. So AA plus XX here, female, AA plus XY, male. And this is the type of sex determination where XY male, XX female determination example is humans and Drosophila. This is an NCERT diagram. Now, what about human beings? Since we occupy apex of the animal kingdom. In humans, originally, male or female determine the sex. No doubt, it is male. So, male determine the sex of the offspring because males are XY. So, two types of gamete. X of male couples with X of female, it is female. Y of male couples with X of female, it is male. So, that is why the male determine the sex of offsprings in human beings. I think that aspect is clear. Now we can proceed to next page. Female heterogamety. That means female will be able to produce two types of gamete. First one, here we can find Z male, Z0 female. So parents will be A plus Z, A plus Z0. Gamete will be A plus Z, A plus Z and A plus 0. So A plus Z, A plus Z, that is A plus Z male. Because female heterogamety we are discussing. A, Z0, it is female. So Z male, Z0 female, sex determination typically seen in butterflies and moths. Examples important. Match the following, etc. Now, second type, female heterogamety, Z male, ZW female. So A plus ZZ, A plus ZW. Here, A plus Z gamete. Here, A plus Z as well as A plus W coupling, A plus ZZ male, A plus ZW female. So that is Z male, ZW female method example is birds. This is also NCERT example. So example is important, match the following, etc. Theory fill in the blank usually. One question is sure for NEET and theory because purely NCERT based. I think it is clear. Now we can proceed to this one. Some other examples not in NCERT came for different exams previously, many years back. One is melandrium. Melandrium is a that is dioecious plant. Dioecious angiospermic plant. Similarly, second one, it is Dioscorea. That also an angiosperm plant. So when we come to melandrium, melandrium is a dioecious angiosperm plant. XY is male, XX is female, just like our type. XY male, XX female. Second is Dioscorea. You may be remembering in biomolecular you study diosgeny, connected with the medical aspects. Then, in Dioscorea, male is heterogametic, female is homogametic. So in all this case, we can find there will be variation within genus or between species. So commonly this way we are meeting exceptions, variations are possible. I think that idea is clear to all of you. Now, the famous example of sex determination honeybee, haplodiploid sex determination. Honeybee, Apis, here you can find Apis cerna, subspecies indica, that is in India. Honeybee cultivation is called apiculture. Apis means viola written here, so it will be easy. Now, the peculiarity is female is diploid with the chromosome number 2 and 32. Male is haploid with the chromosome number N equals 16. So female only meiosis can take place. Male side haploid only mitosis can take place. So when we come into female side 32, so you'll be getting gametes of 16 chromosome each haploid. Male side only mitosis, so chromosome number is 16 itself haploid. Now what happens is, if this 16 couples with this 16 or any other gamete from here, female side couples with the male, it will be producing 2n, that is 32, then it will be female. Suppose male gamete is not able to fuse with the female gamete, it will be wasted. And what about the fate of the female gamete if it is not able to fertilize with male gamete? Female gamete will undergo parthenogenesis, development without fertilization. And since no fertilization, haplonumber chromosome in female gamete is 16, that will be parthenogenetically developing into male itself. This is a special mechanism seen in honeybees. Event is called haplodiploid sex determination system. Simple, prominent for exam point of view, theory understand. These are the honeybees 3 type drones. Male N equals 60. Female queens, both the females 32. Female queen is 32. Worker also 32. I think this page is clear to all of you. Now we can proceed to next page. A famous statement, theory entrance, theory for commenting entrance, true or false. In honeybees, males do not have father because that male honeybee comes from female side due to parthenogenesis. So do not have father and thus cannot have son, but can have grandfather and grandson. I think idea clear because male gamete not able to fuse with the female gamete, male gamete will be wasted. Female gamete will develop into a male offspring. You cannot use the term offspring, male progeny with N equals 60. Okay. Suppose female gamete able to fuse with male gamete, then it become diploid. That is why in honeybees, males do not have father because it comes from female side and thus cannot have son. Male gamete either aborted or will be fusing with the female gamete, then it become diploid female. But can have grandfather and can have grandson. It's a typical example. I think idea is clear. Now comes sex-linked inheritance. In 1910, Thomas Hunt Morgan, T.H. Morgan, discovered sex linkage. Last episode, you studied term linkage also given by T.H. Morgan, previous question. Morgan worked in Drosophila fruit fly. Many experiments he done. In this context, 3 specific experiments. One, eye color, that is Drosophila in connection with eye color in Drosophila, where 3 types of experiment Morgan performed. First one, red-eyed female with a white-eyed male. Second one, red-eyed hybrid female because red eye is dominant, crossed with the white-eyed male. Now, white-eyed female crossed with the red-eyed male. After a series of experiments, Morgan reached into an assumption or a conclusion. Eye color gene is linked to sex and is seen on X chromosome. Different crosses he made in Drosophila, like red-eyed homozygous, red-eyed hybrid, white-eyed female, crossed with the white-eyed male, red-eyed male, respectively. And Morgan was able to find eye color gene is linked with the sex and is located on X chromosome. There is a pattern for it. That is, here you can find father to daughter to grandson. That inheritance is called diagyny. Second is mother to son to granddaughter, that is diandry. And this type of cross, that is like a set. This type of cross is called crisscross inheritance. Father, daughter, grandson, diagonic. Mother, son, granddaughter, diandry. So crisscross inheritance. I think it is clear. We can proceed to next page. In this page, you can find 5 theory question, 5 neat model question with the key. So what all things we learned? You can just pass through in this episode. Principles of inheritance and variation, sixth one, topics, 5 plus 1. Then initially we identified sexuality came when and where. What are suggestions? After that, why sexuality developed, adaptation, and what is sex determination. After that, we entered into chromosomal, most acceptable theory, chromosomal genetic base of sex determination, of which Hermann Henking, 1891, spermatogenesis of certain insect. Sperm will be carrying a specific structure. Only 50% sperms have that, and 50% lack it. He called it as X body, later it become X chromosome. Now types, male heterogamety, female heterogamety, that is 2 types of gametes will be produced. Male heterogamety, 2 types. That is first page. X0 male, XX female. Example is grasshopper. Second is XY male, XX female. That is human beings and Drosophila. Here the diagram you can find. And in humans, originally sex is determined by male only because XY is there. Males are heterogametic. Females are only one type of gamete, X. So humans, male determines the sex. Now comes female heterogamety. Z male, Z0 female. Then example is butterfly and mouse. Female heterogamety. So female will be producing 2 types of gamete. Second is Z male, ZW female. And typical example is birds. This is an NCERT diagram. And some exception like melandrium, diascoria. Melandrium, it is XY male, XX female. Diascoria, it is male is hetero, female is homogametic. And there will be variation between the species, even in determination. Now famous honeybees, haplo-diploid sex determination. Female is diploid 2 and 32, male is haploid N16. Female side meiosis will take place, male side only mitosis. Then one gamete fuses with the male gamete, then it becomes 2 and 32, again female. Suppose male gamete is not able to fuse, it will be aborted. Female gamete will be parthenogenetically developing into male. That is a special mechanism seen in honeybees, haplodiploid mechanism, sex determination. Now comes sex-linked inheritance. Thomas and Morgan, 1910. Linkage also given by him. And experiments in Drosophila. Here you can find that is red-eyed female, white-eyed male, red-eyed hybrid female, white-eyed male, white-eyed female, red-eyed male. This way crosses is done, and he was able to observe that is sex. Eye color gene is linked with the sex and located on X chromosome, and there is a pattern of inheritance. It is father to daughter to grandson, diagonic, then mother to son to granddaughter, diandric, and this type of inheritance is called crisscross inheritance. Father to daughter to grandson, mother to son to granddaughter. After that, we're done 5 theory question, 5 neat model question with the key. If you like this episode, subscribe all, like it, share it, comment on it. We can meet in the next episode of genetics. For time being, thank you. We can meet later. Take care.